In the digital age, data is king. We generate massive amounts of data every day, from selfies and social media posts to scientific research and business records. Traditional storage solutions like hard drives and optical disks are reaching their limits in terms of capacity and longevity. Hey tech enthusiasts, welcome back to TechFlix. Today, we're diving into the fascinating world of cutting-edge technology, DNA-based digital data storage. Yes, you heard that right. We're talking about storing digital information in the very building block of life, DNA. Buckle up, because this is one of the most exciting frontiers in data storage, and we're about to break it down for you in this video. In a surprising revelation, researchers have put forth a concerning prediction. Within the next 3 years, the world's data centers may struggle to accommodate even half of the massive volume of data generated by internet users. However, this doesn't necessarily spell doom for the internet by 2026. The solution may lie in a remarkable proposition. Instead of relying solely on conventional hard drives, tech companies could transition to using synthetic DNA for data storage. It might sound implausible, but researchers contend that DNA strands possess the remarkable capability to store vast amounts of digital data. In fact, they argue that synthetic DNA offers several advantages over modern-day data centers. Can storing data on DNA really be done? Storing data on DNA is indeed a viable concept, and it's not a novel idea. DNA, renowned for its role in housing an organism's genetic information, has been considered for digital data storage before. The initial proposal for using synthetic DNA to store digital data dates back to the 1980s. In a significant milestone, around 3 decades later, in 2012, a team of scientists at Harvard University successfully encoded a 52,000-word book into DNA fragments. Remarkably, they achieved the capacity to store a staggering 1.29 petabytes, equivalent to 1,280,000 gigabytes of data on every gram of DNA employed for this purpose. Nonetheless, despite promising developments in DNA data storage, several inherent limitations hindered its widespread adoption as a mainstream solution. Over the years, numerous scientists have endeavored to address these challenges, yet some obstacles have remained unresolved. One significant hurdle stems from the resource-intensive and costly nature of encoding data onto DNA and subsequently extracting it. The current method employed for reading data from DNA, known as polymerase chain reaction, has proven problematic. During PCR, a matching prime is used to locate and bind to the target DNA strand containing the stored data. Subsequently, this DNA is replicated millions of times to enable the system to locate and retrieve the desired file. However, a fundamental drawback is that each reading of the data deteriorates the quality of the original DNA. This degradation makes it challenging to read multiple files simultaneously and introduces errors into the copying process. In essence, reading multiple files concurrently necessitates multiple prime pairs working simultaneously, compounding the risk of errors in the replication process. When considering DNA, most people associate it with life rather than computers. However, DNA itself is a code comprised of 4 letters used to convey information about an organism. DNA molecules consist of 4 types of bases or nucleotides, each represented by a letter: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases serve as the fundamental building blocks of all DNA code, serving as the instruction manual for constructing every living entity on Earth. DNA synthesis is a well-understood technology that has found widespread applications in fields such as medicine, pharmaceuticals, and biofuel development, among others. This technique arranges the bases in specific sequences of A, C, G, and T to create various patterns. These bases coil around each other in a double helix structure, forming the DNA molecule. The arrangement of these letters into sequences constitutes a code that provides instructions for an organism's development. The entirety of DNA molecules constitutes the genome, essentially the blueprint of an organism's body. Through DNA synthesis, which involves creating DNA molecules from scratch, researchers can specify long sequences of the letters A, C, G, and T, and subsequently read these sequences. This process is conceptually akin to how a computer stores binary information. Building upon this concept, researchers have successfully encoded binary computer files into DNA molecules. While this method has proven its feasibility, the process of reading and writing DNA-encoded files currently consumes a considerable amount of time. Adding a single base to DNA takes approximately 1 second. Writing an archive file at this rate could span decades. However, ongoing research is focused on developing faster methods, including massively parallel operations that can simultaneously write to numerous molecules, thus accelerating the process. The systems provide a precise blueprint for translating the binary language of zeros and ones into sequences of 4-letter combinations using the genetic alphabet of A, C, G, and T. Notably, the systems also facilitate the reverse process, decoding the DNA data back into binary form. It can adapt to various methods of DNA synthesis, making it a versatile tool for data storage. However, DNA-based storage introduces unique challenges compared to traditional digital systems. Error rates during DNA synthesis are notably high, stemming from different sources than those in the digital realm. While digital errors involve flipping zeros to ones or vice versa, DNA errors often result from insertions and deletions. For example, the intended sequence of ACGT may encounter issues where an A doesn't register, causing a shift in the letter sequence or even repeated letters like AAA. To address these challenges, researchers incorporate error detection codes to validate the stored data. During the conversion back to binary, the software checks for code consistency. If discrepancies arise, it employs corrective measures by adding or removing bases until verification succeeds. As data generation continues to surge exponentially and traditional digital data centers face challenges in terms of cost and sustainability, DNA-based storage emerges as a promising solution. While this technology requires new tools and innovative approaches, it offers the potential to store vast archives of valuable data within minuscule collections of molecules, revolutionizing data storage in the future. In conclusion, DNA-based digital data storage is a cutting-edge realm that holds immense promise. It's not just about storing data, it's about preserving our history, culture, and knowledge for future generations. So, keep your eyes on this space, because the future of data storage might very well be written in the language of DNA. Thanks for joining us today on this incredible journey. Don't forget to like, subscribe, and hit that notification bell to stay updated on all things tech. Until next time, stay curious and keep exploring.